Okay, I'll try to explain how interferon exerts its antiviral response in a little bit of detail. Let's say the first scenario, which is you have virus infecting any cells, any somatic cells, cells that are not gamete cells. So once these cells are infected with virus, the virus does its replication as what we explained in topic 1 to topic 4 earlier, and then it produces its new viruses. At the same time, when infection happens, you remember that each of these viruses, regardless of what kind they are, that will always produce replicative intermediates. These are conditions where the genome of the viruses presented itself in a way that is very foreign to the cell, like double-stranded RNA, single-stranded DNA, or unique motif combination between DNA and RNA. Those are the cells, things that the cell have never seen before. And these will activate the interferon regulatory factors and makes those infected cells produces interferon. There are two classes of interferon, class 1 and alpha and beta, and class 2, which is interferon gamma. Okay, depending on the cells, the type of the cells, these are the kind of interferon that they produce. So what happened to this newly produced interferon? It can either affect the infected cell itself, back on the infected cell itself, or it will also affect the neighboring cell that is not affected or infected by the virus yet. So what happened when interferon, what happened to this interferon is the interferon will bind to receptors on either of the cells, whether the infected cells, the ones infected earlier, or the still healthy neighboring cells. Once interferon binds to the receptor, it will activate the JAK-STAT interferon pathway, and this will produce interferon responsive elements and produce three kinds of proteins. So you see that the kind of proteins are what we call PKR, OAS, and RNase L. So what PKR would do is it will inhibit mRNA translation. Basically, it inhibits the eukaryotic translation factors. So that means there's no more mRNA being translated, regardless whether it is viral or host mRNA, because the virus also depends on cellular machinery to translate its protein. So that's the function of PKR. OAS is another protein which stimulates the activity of RNase L, which actually is a ribonuclease, and ribonuclease, as the name suggested, is actually acting on any mRNA and digest mRNA. So basically what happens is when you have interferon binding to any of the cells receptors, it will activate this condition where it's no longer suitable for protein synthesis to happen. And so if you can imagine this kind of neighboring cells that is still healthy, infected with a new virus, when the virus gets inside there, it's no longer in the condition that will support this protein synthesis, and virus infection stops. So this is how interferon exerts at least an early stage of antiviral response inside your cells.